Okay, so welcome everyone to the workshop for software for synthetic biology workflows, or how to improve productivity and your impact. So during this session, this is a regular Zoom call, so you have the ability to raise your hands or to type questions in the chat. So please feel free to do so during any of the presentations. We'd be happy to take your questions during the presentations. We'd like this to be a little bit more interactive and allow for time for discussion. So please go ahead and do that if you have questions that come up. So this session is going to be organized into 4 parts. There's going to be a presentation on a modeling workflow for genetic design given by myself, a presentation on a production workflow for the design of libraries of genetic constructs given by Beal, a data management workflow for genetic circuit characterization experiments will be given by Gonzalo Peña, and a test and learn workflow for high throughput laboratory science, which will be presented by Bryce. So the first presentation, as I said, will be given by myself on a modeling workflow for genetic design. So in this presentation, I'm going to be talking about 2 projects. The first one is a synthetic biology knowledge system. This is a project funded under the NSF Harnessing the Data Revolution program. And basically, it's an interesting project because it involves a wide group of people. We have 6 PIs on this project. We have people that are experts in text mining and machine learning, social science, and biology and library science. So in this project, what we're doing is we're developing a knowledge system that stores information from literature about synthetic biology projects. And so in this project, we have developed 2 pipelines, one for mining this information from text and another for mining it from data. So in particular, we began with electronic versions of all of the ACS synthetic biology articles represented in an XML format. We extracted the metadata from that and encoded that in SBOL and deposited it into the SBKS system. We've also analyzed the text and using machine learning and text mining techniques and auto-annotated things like which organisms are talked about, which genes, which proteins, small molecules, etc. We then took these annotated versions of these articles and had them analyzed by domain experts who would then look at that and correct the annotations. And then this would be used to refine our machine learning techniques. The end result would be that we would have annotations on each of the articles about what the articles are about, what genes do they talk about, what chemicals, what proteins, what organisms do they talk about in using in their projects. We've also got the social scientists working on analyzing a broader swath of literature from PubMed, and he's looking at, for example, collaboration networks within the synthetic biology community, as well as the publication of ethics and related topics in synthetic biology. So in the data mining pipeline, what we've been doing is we've been analyzing sequences found in the articles, in particular in the supplementals provided with the articles. So we have mined the sequences out of these supplemental documents, and then we have put them into a tool developed at BB and Raytheon called SynBioHub, which does auto annotation of these sequences to annotate them with parts, and then this information is linked to the publication where they came from. So in the text mining pipeline, this is an example of one of the results. This shows a word cloud of the entities that have been found in ACS synthetic biology articles. As you can see, E. coli is an organism that's found in a lot of publications, as is GFP, Cas9, yeast, etc. And so there are a lot of terms that we've been able to identify to help people who are interested in finding articles about particular topics. Another aspect of the text mining pipeline is topic modeling. So we've been analyzing the papers and then identifying what topic these papers are talking about. So after doing topic modeling, we found that there were approximately 5 core topics published in ACS synthetic biology. We have genetic circuit modeling papers, genetic circuit design papers, strain construction papers, biosensor and protein related papers, and metabolic engineering papers. And they each represent approximately 1/5 of the literature in ACS synthetic biology. On the data mining side, we've developed a workflow for analyzing sequences and annotating those sequences. So in this workflow, we had our domain experts analyze a number of toolkit papers. So these are papers that describe genetic circuit libraries They then extracted information about these genetic circuit libraries and put them into Excel spreadsheets for each of these genetic circuit libraries. And you'll hear more about these spreadsheets, I think, in the following talk from Jake Beal. So we basically build up these spreadsheets of libraries of parts. We then used a tool that we developed called Excel to SBOL, which translated the Excel representation into SBOL, or the synthetic biology open language. And then this created an SBOL representation of these library of parts that we could use for our auto annotation. We also took all the supplemental files, these are PDFs, GenBanks, text files, SBOL files, etc., and we analyzed those to try to find DNA sequences. And so we built a tool that would extract from these files anything that looked like a DNA sequence, basically any sequence of the characters A, G, C, T. Then we had a collection of FASTA files for each of the sequences that we're able to extract. And then we sent these FASTA files into this SynBioHub tool, coupled with the library of parts that we'd built. And then we auto-annotated the sequences to identify which parts were being used in which sequences from which papers. These annotated sequences have then been deposited in the SBKS data store and coupled with the papers from which they came from. So this would allow you to do things like identify a paper that talks about, say, a particular coding sequence or promoter that you're interested in. So here's an example of some of the data that you can mine out from this information. So these are the most common annotations from the ACS synthetic biology supplemental sequences. So the first column is annotation, the most common annotations regardless of the type of part. So for example, a ribo-J was the part that we found the most often in these sequences. The next thing is a terminator, a specific promoter, etc. And then I also have sort of the top hit lists for divided down by promoters, coding sequences, terminators, and other types of parts. One thing I want to strongly emphasize and strongly highlight here is the fact that we began with the files in the supplemental, which were composed of PDF files, SBOL files, GenBank, and FASTA files. And the vast majority of the files that we had were PDF files, and the vast majority of the sequences were extracted from those. So we mined sequences from all of these, but the sad note is that the sequences that came out of PDF files were never actually successfully annotated. So it was very difficult to extract meaningful and useful sequences from the PDF files. And in particular, we had lots of problems with the fact that the sequences, for example, would have carriage returns in them, and it'd be hard to know where one sequence ended and another began. So this led to a lot of problems, and this is yet another reason we want to strongly advocate that if you're publishing articles, you need to publish the sequences in a standardized format, FASTA, GenBank, or preferably SBOL. Yeah, David, do you want to unmute yourself and ask your question?
Yeah, just real quick. Last time I looked at ACS SynBio, I thought they required designs to be deposited in an SBOL format. Is that something that's just too new and that you weren't able to take advantage of?
The key word in your question there is required. They're recommended. And I'm not sure, I've had trouble finding that recommendation on the website of late. It used to be in every publication you submitted, you got an email telling you that you were recommended to do that. But very few people would follow through because it was not a requirement. I think this is a big problem. I have talked to the editors and managing and the consulting editors with ACS SynBio, and unfortunately we didn't get much headway in strengthening that requirement. I mean, their requirements of the publication do say that if you build something, you have to share your sequences in some way. But they don't really precisely say whether it can be just in a PDF file or in a standard format that's usable. And I really think that they need to consider strengthening those requirements, because when I publish an article in ACS SynBio, I mean, I spend a lot of time trying to make sure and prove that I have like rights to the figures I use. I mean, similarly, as you're publishing an article, I think, spending time to make sure that you've deposited your sequences somewhere, it does not seem to be too heavy a lift. So I really hope that we can reopen this discussion with ACS to get them to more strongly enforce the policy of depositing sequences in an electronically readable format. And I think this data goes to show that without doing so, it's next to impossible to get that information out in any meaningful way. I mean,
this is on the peer reviewers too.
It is on the peer reviewers too, right? So whenever
the peer reviewers say, put it in or I don't approve, that's the best first forcing function. Leaving it to the editorial staff is the fallback.
Right. So I'm not asking the editorial staff to do the checking per se. I mean, the reviewers need to check it, but it does need to be a rule, and it need to be a rule that's enforced, not just a recommendation. And there has to be clearly delineated that the formats need to be in machine readable formats. PDF is not a machine readable format for sequences. And so I think that this would be a huge benefit to the community if we could enforce this. So getting off my soapbox. And the last thing I was going to say about this is that one of the things we came down to, because we've annotated these parts where we have successfully annotated sequences, what I'm showing here are the top 10 publications where we have found some use of parts from the MIT BU Cello Part library. Okay, so the second part of my talk, I'm going to be talking about an automated model generator workflow in which we take parts from a SynBioHub, a data repository, construct designs using the SBOLCanvas tool, and then we update these designs with additional information also mined from SynBioHub to produce computational models that we can then simulate and then further share on SynBioHub. So I've got several little short demos to show you about that. So this first one here, this video is showing the SynBioHub data repository for the NSF Living Computing Project. And so when you're trying to build what I'm going to do in this demo is I'm going to be building a part library that I can then use in my design. So I'm going to go onto SynBioHub looking for the parts that I want to use. So I'm going to create a special sort of part collection for these parts that I want to use in my design. So the first thing I did is I logged in, and now I'm going to create a part collection and where I'm going to drop in the parts that I'm going to be using in my genetic design. So this is a collection of my parts that I've created now on SynBioHub. So this is a private part collection, and you can kind of think of it kind of like a shopping cart. I'm basically building a shopping cart for the parts that I want to use. So I'm going to be using parts from the Cello E. coli parts library. And so I'm going to be looking for various parts in this library that I want to use in my design. So the first one that I want to use is the LAC-I sensor. And so this is going to produce the LAC-I compound. And so I'm adding that to my parts library. And then I'm going to go back to the set of parts, and I'm going to find some of the other parts that I want to use in my design. And so I also need a TED-R sensor. And I'm going to add that to my parts collection. And so I think I'm actually going to skip ahead on this one a little bit because I just keep doing the same thing. I go find my various parts. So here I'm adding YFP reporter. I add that to my parts collection. And then when I'm all done, I've got a parts collection. Now this is not copying the parts. These are just kind of linking. I've just built a cart of parts that I want to use. So they're easier to find when I go onto the next part of my project, which is I'm going to design a genetic circuit. So in the next demonstration, so in the next video, I can scroll ahead. Come on. I'm going to use those parts in a design of a genetic toggle switch using Cello parts. So in this video, I'm going to show.
In this video, what I'm showing is a tool called SBOLCanvas. You can find it at sbolcanvas.org. And so first you navigate to that location. I'm going to give the name of my design. It's going to be Cello toggle switch. Actually, that was the ID. The name is Cello toggle switch. And then I can give it a short description. And so this is my Cello toggle switch. Of course, you can use more clever descriptions than that. And so the first thing I'm going to do is I'm going to add one of the sensors. So I'm adding a DNA component. It's an engineered region. And then I'm going to import that from SynBioHub. So using the import component, I'm scrolling through. Well, first I need to log in because I need to find my part collection that I had just built. So I'm logging in. And then I can scroll down and find my parts, the collection I just built, and then I can find the TED-R sensor I want to use. And I've added that to my canvas, as you can see here. So this TED-R sensor, it produces the TED-R protein. So I'm going to add that to my canvas. And I'm going to indicate that this is the TED-R protein by filling in the ID over there. And then I'm going to indicate that it's generated from this coding sequence. So I select my two parts using Command on an Apple,
and select the arrow I want to generate between them. You might also want to customize the color so it's easier to see. And so I'm going to make the color of the coding sequence in the protein the same. So I'm making these both blue so that it's clear that the coding sequence and the protein are related to each other. So the next thing I'm going to do is add a small molecule. This small molecule binds with TET-R in my genetic toggle switch. The small molecule is called ATC. So ATC and TET-R bind to form a complex. So I'm adding my association node to indicate that it forms a complex. And I'm adding the complex into my schematic. This complex is the TET-R ATC complex. And so ATC goes in and TET-R goes in, the TET-R protein, and they form this complex. Okay, so the next thing I'm going to do is add another transcriptional unit. So this is the first half of the genetic toggle switch. So this is composed of 2 promoters and an engineered region. So the first promoter is going to be a promoter that's repressed by TET-R, the p-TET promoter. And the second promoter is going to be one that is repressed by one of the cello proteins. In this case, it's going to be the PHLF promoter. So it's repressed by PHLF. And then I need an engineered region here that is going to produce the other protein, which is AMTR. And I've added that to my canvas. Now, this is actually a composite part. You can see the little dashed arrow underneath it to indicate that. You zoom in, you can see that it's composed of a riboJ, an RBS, a coding sequence, and a terminator. So you can pop back out. So as I said, TET-R represses the p-TET promoter. So I can select both of these or command select both of these and then indicate that there's an inhibition arc between them. And to show that these are related, maybe I want to fill in the color, make sure that they have the same color. Similarly, AMTR produces the protein, the AMTR engineered region produces the AMTR protein. So I can select these 2 things. First, I'm going to label it as the AMTR protein. And indicate that this produces that. And then the last one I'm going to put on this half is a reporter. And so the reporter here is going to be a promoter and an engineered region to produce YFP, or yellow fluorescent protein. So the promoter is going to be the AMTR promoter that's repressed by AMTR. And then the reporter is the YFP reporter. And so that was just showing what was inside the YFP reporter. And the AMTR protein represses the PAMTR. Now this is half of the design. Well, actually, I'm going to add in the YFP protein. And like that, going to add a little bit more color to indicate the relationships that we have here. So this is going to be colored red to indicate that they are all related to each other. And then lastly, of course, this will be colored yellow because it's the yellow fluorescent protein. So you can see that SBOLCanvas is also a useful diagramming tool. Now this is just half of the design. I'm not going to go through the other half. Like a usual cooking show, you've got the pre-baked one in the oven. So this is the other half of the toggle switch for that design. And so this is the completed toggle switch design using SBOLCanvas. So the next part of this is I'm going to show how you can do a computational model of this design that I've just shown you. Now I'm going to show this a little bit differently. I took all the interaction arrows out Instead, I'm going to let the modeling tool actually add this interaction information that is stored in the SynBioHub repository for you, basically indicating what proteins are produced and repressed per motor. So this is just showing the 5 transcriptional units. So in order to do this, first thing I have to do is I need to share this design on SynBioHub so I can download it into my other software tool. So I'm going to take this design and upload it to SynBioHub. So I need to select on the file option, and I'm speaking a little bit ahead of my talking there. So select upload to SynBioHub, select a SynBioHub to upload to. I'm going to create a collection to put my design into, which I'll call My Designs. Collection of my designs. And create that collection. So now I select that new collection I've just created, and I'm uploading this design into that new collection. And this is going to enable me to then download it into another tool. So I can go to SynBioHub and now see that indeed it has been uploaded. If I go to my designs and go look for the module that I've just uploaded, which is called Cello Toggle Switch. You can see it's got the 5 transcriptional units that I had built in SBOLCanvas. So now I'm going to move to a tool called iBioSim, or the Intelligent Biological Simulator. And I've got a project here where I'm going to download the toggle switch model into it. So I'm going to log into the SynBioHub so I can fetch it from my private repository. And then I'm going to select the design, and it's going to download it into my project. And now I want to generate a computational model for this design. So I'm going to go open up the file of my library of 5 transcriptional units. I'm going to highlight them and select Generate Model. Give the name to my model. Where do I want to get, this is where I want to get the interaction and modeling information. It's from that SynBioHub instance. So it's fetching all the interaction information in this design. And it's building a computational model and encoding it into the Systems Biology Markup Language, a data standard for modeling, or SBML. So what it's done is it's determined that there's these 5 different circuits. It's built models. So see, it's added the interactions that AMT and LAC-I repress the circuit that produces the PHLF protein. And I'm going to now instantiate the entire design into an environment module, which is going to simulate essentially the experimentalist that's adding the IPTG and ATC and observing the YFP output. So this is an environment model that I pre-built. So I'm adding the toggle switch in to the model. And then I need to connect in the inputs, which are IPTG and ATC, and connect out the output YFP. On the right, I have something called a PetriNet, and it's what's going to actually stimulate the different events. So this PetriNet event here is, if I can click on the right spot, is going to set IPTG to a high value after 2,000 seconds. It's then going to set it to a low value after 2,000 more seconds, and then set ATC high at 2,000 seconds more, and then finally set it low 2,000 seconds after that. So now I create what's called an analysis view. And I can perform a simulation. I can do ODE or stochastic simulation. I'm going to do ODE in this case. I'm going to simulate for 10,000 seconds at a time step of 10. And I am going to do abstraction on the model, make it a little bit faster. And I run my simulation. And now we can look at the results. ATC, IPTG, YFP, and you can look at the simulation result. And it looks kind of like a toggle switch. It's a little bit uncertain in the beginning because we didn't set an initial state, so it doesn't know if it wants to be high or low, but then we put the set input, turns it on, take the set input, IPTG away, it stays on, add ATC, it goes low, take ATC away, it stays low. So this looks like it's operating like a nice toggle switch, or is it? So the next demo is actually here.
In the last
So
demo, we demonstrated
in this demo, it's basically creating a model using a different technique. So in the last demo, I built the model using default parameters. In this one, I'm going to be using the characterization parameters that were measured in the laboratory at MIT and are stored with the Cello library. So what I'm doing here is I'm going to download again from SynBioHub into a new project, this design that I've made. I'm going to add that to the project, and then I'm going to regenerate the model, but this time I'm going to select Cello modeling. So rather than using default parameters in the model, it's going to use the actual parameters that were measured in the laboratory for each of the gates. So I'm going to download that information from that SynBioHub. And it's pulling that information across. It's done. And here's the model. So now I'm going to add that circuit into again. I don't need to go quite through all of that, maybe. And build another analysis view. The time course is a bit different here with the real parameters, so it runs for a little bit longer time. And rerun the simulation. And this time we look at the results. And unfortunately, it doesn't look like a toggle switch anymore. So what you see here is at the beginning, it actually floats to a high value because the high part of the circuit actually is a bit stronger than the low part. It can get set high and it can hold its high, but then when you reset it, it'll go low-ish, but it doesn't go completely low. And then when you take the reset away, it floats back high again. And so this is just representing the fact that in this design, that the circuit that holds it high is stronger than the part that holds it low. When we use the actual parameters, we find that this actually isn't going to behave exactly the way we want. So this shows the importance of using real characterized parameters when you're doing your modeling. And then in my very last, little demo here.
This demonstration, we're going to show
what I'm showing is how you share the model when you're all done. So once we're done with all our modeling work, maybe you want to share it with a colleague so we can upload that to SynBioHub. I'm going to put it into a new collection called My Models. And I've uploaded that. So now you can go to SynBioHub again, refresh the page, go to My Models, and you can see that all the modeling information I created has been stored there. You could download it into another iBioSim if you wish to simulate, or other SBML supporting tools. It includes here, for example, the simulation plot that you had seen me produce. So that concludes my demonstrations of this modeling workflow. So I just want to conclude with some thanks. So the SBKS project, as I said, it's an NSF HDR Ideas Lab. So it's a very exciting project with collaborations from UC San Diego, WPI, University of Illinois, NORC, and VCU, and my own group that spans the University of Utah and University of Colorado Boulder. And this is the current research group that is working with us. My PhD students and undergrads that are working on these projects. I want to thank the NSF and DARPA for funding this work. Microsoft Research has provided some server where we store things like our SynBioHubs and SBOLCanvas for people to use. And there are many students have been involved in the development of these projects. So this is a list of the people that have since graduated that have worked on SynBioHub, SBOLCanvas, and a previous tool called SBOLDesigner, and the iBioSim tool. And I'd also like to thank the SBOL Industrial Consortium and the Go Summer of Code for funding some of these students in the past. And last but not least, here are some links where you can get some additional information. I know that I'm right at time. I saw that there were some questions in the chat. In order to keep on time, I'll be happy to continue to answer questions in the chat in parallel while the other talks are going on. But with that, I think I'm going to hand it over to Jake Beal to take on the second workflow.
All right, is my screen coming through okay?
Yes.
Okay. Hi, folks. I'm looking forward to sharing with you something about a workflow that we've been using in the iGEM engineering committee for collaborative design of sequences. So Chris was talking about something that, it could be collaborative, but could also be sort of walked through by one person who's bringing together a whole bunch of different pieces. On the iGEM engineering committee, we've had a somewhat different challenge where, because there are a lot of people in a lot of different places, we actually have to do everything collaboratively. You can't have just one person designing a sequence by themselves because there's a lot of different people who want input on what that sequence is and whose input is needed. Because there's a lot of people with different pieces of the puzzle of the things that we're trying to investigate. So we need to have collaborative design in an online sort of context where you've got a whole bunch of people collectively editing what in our case turns out to be what I think of as the lowest common denominator of biological design, a spreadsheet. We're designing fairly complex library builds in this spreadsheet, which then we have a pipeline that's SBOL enabled for taking those designs and getting them into FASTA files that we can upload to a DNA synthesis company to get built in support of our experiments. And of course, as soon as we upload it, what anybody who's ever ordered sequences knows is as soon as you upload the sequence, you find that there are manufacturability issues that you hadn't realized, and the synthesis company rejects your sequence and you need to go back and redesign, in this case, with your dozen collaborators scattered over a bunch of continents. So it was important to us to make this bit where we go from that sort of whiteboard sketch of the plan as captured in an Excel sheet into a whole bunch of FASTAs for different things that we're ordering as smooth and fast as possible, so that we can spend all of our time over here at, around the virtual whiteboard figuring out what we're doing and how we need to adjust it, rather than waiting for somebody to spend an hour putting all of the adjustments into all of the individual designs for each of the different plasmids that we're looking to build. So let me tell a little bit about the context of this with the engineering committee. The iGEM engineering committee, iGEM is the International Genetically Engineered Machines contest and jamboree, where hundreds of teams of undergraduates and high schoolers all around the world basically spend their summer doing synthetic biology projects and then get together and present them to each other. The engineering committee is aiming to support this with a group of volunteers doing three things. Educating the teams participating on iGEM on good synthetic biology engineering practices, establishing and disseminating strong foundations of measurement and calibration protocols and standards so that they have good units to build on and share and be able to build off of one another's work, and improving the biological devices and models of those devices and their behaviors in order to make it easier for everybody to engineer. We've got a lot of people on this committee. There's more than 50 members from 40 organizations all around the world, 5 continents, 18 countries at the moment. And one of the things that tends to happen within this committee and its predecessor, the measurement committee, is organizing interlaboratory studies to test out the things that we want to inflict on these students around the world. Because if you can't get something reproduced in half a dozen labs of professionals, you really don't want to hand it over to a bunch of undergraduates and high schoolers and see if they can make it work too. So when we're designing interlaboratory studies, one of the key bottlenecks is committee design of genetic constructs. Because you've got a lot of people with a lot of different perspectives and a lot of information you want to include here. So let's go back to this sort of unifying picture. And I want to focus first on the place where we're doing our design to tell that part of the story. And we're basically just working in Google Sheets. This is a terrible design tool if what you want to do is, sequence optimization and so on. But if what you want to be doing is basically saying, I'm taking a bunch of parts that I want to put together in a bunch of different ways because I want to test things out about them, it's a good place to put together sort of whiteboard sketches of an experimental campaign. And as a collective, cloud editing platform, it's a great place for us to be, working together on these sorts of things in a collaborative fashion. You can see who else is editing. It's all live. It's the cloud. It's good. So we're writing this all down in essentially 2 different tabs of a Google Sheet with some other stuff backing it, where what we're fundamentally doing is planning experimental campaigns in terms of, I want to put together these combinations of parts into these plasmid backbones, and sketching that out at sort of a whiteboard design level. So for example, in here, one of these lines here, this line about these, fluorescent proteins, small batch, is saying we're going to put this into this p-open v3 plasmid backbone. First we're going to put this upstream 5' and then one of 2 promoters, and then some spacer material around a ribosome entry site, and then there's some more parts going on off to the other side, a coding sequence, a terminator. Other places we're saying, like, I want, for my collection of fluorescent proteins, we're going to try this whole set of different fluorescent proteins, all these different options, into which I'll then plug into another spot here. So we sketch this out in terms of some very sort of human intuitive ways to do this. But because we've defined the columns in a machine readable fashion, we're going to be able to rip the information out of this with some scripts later. Now, all the things being defined in here are either defined on one of these lines or on the other sheet, where we're putting down stuff like, here's, we're going to have some BioBrick scars and the stuff that we're synthesizing. Even though we could synthesize it without a scar, we're saying what would happen if somebody had built this directly with a scar in this particular project. And it turns out the scar is actually sometimes necessary for performance. Wouldn't like that, but it's true. And here's a bunch of the different fluorescent proteins we want to keep in. And over here, off the screenshot, there's the places where the actual sequences show up, etc. So this is all using a format designed for machine readability and human readability in an Excel template that you can find at this Excel to SBOL project. So, okay, we sit around, we talk on Slack, we sketch out our project here, but now I need to go from all these very sort of compact annotations into actually stitching together all the different combinations that are implied by, I want to put all these fluorescent proteins into the different coding sequences with these 2 different promoters up front, etc. So let's talk about how we actually built our pipeline for turning designs into orders that we can then take to the company and get checked out. So we've implemented this as a chain of fairly simple scripts, leveraging SBOL to go from this sort of high-level sketch that we can represent fully all the way down into FASTA files. So there are 4 stages in this. The first is ripping the designs out of the Excel spreadsheet and turning those into SBOL in a particular notion called a combinatorial derivation. That's how you represent a library of different design options. We then expand those sort of whiteboard sketches of a library into what are all of the individual elements of the library by expanding those combinatorial derivations into specific designs where all the variables are filled in. Then we need to take the sequences of the parts and bubble those upwards in order to calculate the sequence for the full construct. And finally, take that out of SBOL into FASTA so that we've got a file that we can upload to a synthesis provider and find out where we've got problems that they want us to modify the design, et cetera, until we're actually ready. So like I said, we implemented this as each one of these pieces is a separate Python script, and we just chain them together. If we were designing with something else as the front end, like some of the tools that Chris showed, like SBOLCanvas, that you can make these libraries in also, then we could do all the latter portions of that as well from there if we wished. 3 libraries that were key for making this work. There's a great Excel reading library called OpenPixel in Python. Makes it really easy to work with spreadsheets. And then we use the SBOL Python library and this library called Tito, stands for Take Your Turns from Ontologies. That's used for working with some of the ontology terminology for labeling bits and pieces. You know, what is a promoter, what is a coding sequence, et cetera. So let's talk about how we actually did each of these stages. I'm not going to go into too much depth, but I just want to unpack each one and really show in detail what exactly it is that gets done there. So for the first stage, extracting the combinatorial derivations, we're taking this spreadsheet and pulling out of it essentially a machine-readable version of a whiteboard sketch that looks like this. We're going to put all the different constructs in the fluorescent protein small library into the popen V3 plasmid backbone. What are those constructs? If we expand them out, we find that we're starting with a spacer, then one of 2 proteins, then a 5' UTR chunk, then one of a number of fluorescent proteins, because this particular project is looking at some alternative for standard fluorescent proteins, followed by a terminator. And then some of these in turn expand out. So this spacer is made of 2 different chunks put up in front of one another. The 5' UTR is a ribosome entry site flanked on each side by scar sites. And the pieces that I've drawn and outlined, these are, think of it as the variables where we're going to plug in different options for our library. So we've now got a sketch of a library, and in fact, pull it out of this. For this particular project, there were several different libraries that we were constructing all of them. So we've got our set of libraries we want to build. The next thing that we're going to do is take each entry in the library. Here, I'm just focusing on the part of that last one that had the variables in it, and fill in all of the values for those variables that we actually want to get built. So in this case, for example, we've got 2 different promoters and a whole lot of different fluorescent proteins. So we're going to get all the combinations. We're going to have the J23101 promoter with the EBFP fluorescent protein, and the J23106 promoter with the EBFP fluorescent protein. And we're going to have J23101 with MTAG BFP2 and J23106 with MTAG BFP2, misspelled apparently, et cetera, for all the rest of the fluorescent proteins as well. So we get all those combinations out. And now we have concrete specifications. This is the point where we now have no more unknowns, no more variables, but we don't yet actually have a sequence for this component. We couldn't have a sequence for it before up here either, because we hadn't picked what the promoter was going to be or what the coding sequence was going to be. We didn't even know how long it was until we made these choices. So the next thing that we need to do is to stitch all these pieces together. We flatten going up. So for that spacer, it had two pieces. So we take those two pieces, we stick them together. We get this sequence for the whole spacer piece. Same thing with the UTR going down in. Once we have the sequences calculated for each of the pieces, each of the 5 pieces in our construct, now we can just concatenate them all together and we get the sequence, the whole sequence for the whole thing. None of this is terribly complicated. None of this is terribly hard, but we need all the little pieces to get from point A to point B. Let's see, I see a question here. Where are the part names translated to DNA sequences? Do you provide the sequence along the part name in the spreadsheet? There, I'm gonna go backwards for a minute here to, this is where that basic parts sheet, that's where we, in one of the columns off the sides here, we have entries like this that I've got highlighted where the sequences are in that first sheet.
Jake, you have a question. Where are the part names translated to
just
DNA?
answered that. I just finished answering that question. We could try and be more sophisticated than this too, and like put a name and have it go out to SynBioHub or to NCBI to go and grab a copy of the sequence from there, but we didn't do that. We just went and pasted it into the spreadsheet ourselves. So at this point, now we actually have enough that we could produce a GenBank file or a FASTA file that has the actual sequence that we want to get synthesized. But right now we have an SBOL file and we can't upload the SBOL file to the sequence provider we're working with because they require that you submit FASTA only. So we have a little chain of conversions that we have to do. It turns out we can't go directly from SBOL 3 that we're using into FASTA yet. There's not an exporter for that. But we export our SBOL 3 to SBOL 2, and SBOL 2 has an exporter to GenBank or FASTA. It turned out that we actually want to export to GenBank first and then use another converter to turn it into FASTA because the SBOL 2 to FASTA converter was a little bit too smart and too aggressive, and it exported all of the sequences from the file, not only the sequences of the particular constructs we wanted to order. So we needed to put only the ones we wanted to order out into GenBank and then turn the GenBank into FASTA. So, you know, a little conversion chain there. Again, nothing particularly complicated, just a little bit of scripting. And what we end up from all this is we end up with a little chain that takes less than 5 minutes to get from, okay, I think we're ready to try to order, to the order actually uploaded at the DNA synthesis company and a manufacturability response from them. None of this is terribly deep. None of this is terribly complicated, but having that little chain of scripts that lets us go from the place where it's really easy for us to talk about at that kind of whiteboard level, how we're putting the pieces together and what we want the pieces to be. Getting that into the collection of FASTA files really fast and easily means that we spend all of our time on Slack having discussions about, oh, it came back with this. Oh my God, we have to make this shorter than this. Oh, you know, this needs to be longer. We got this problem here. How should we do it? You know, what should we do about this question? You know, all the different experts and contributors talking with one another about how to do our science collaboratively rather than making a decision and then asking a grad student to go sit down at a sequence editor to produce you know, some dozens of sequence files to upload. So we just take that portion out of the loop and get to spend more time on the part that's the collaborative part rather than the wrangling the sequences bit. So in summary, Using simple cloud editing tools plus the PySBOL library, in particular PySBOL 3, let us do some really simple scripting for realizing our designs out of the sort of whiteboard level sketches that are much more intuitive for us to have discussions at. It's a complex transformation, but it was able to be broken down into several very simple utility steps that are likely reusable in other workflows as well. We have been using this workflow for collaborative design and ordering. The first interlab is running right now. And where we're going from here, we want to start incorporating, adding some more scripts for design rule checking into that workflow, because there's, checking for illegal cut sites or things like that are things that we should be able to do. As we're using it, we'll keep pushing around bits and pieces of the workflow and improving the tools. One of the things that's coming up is we want to incorporate build planning in there too, rather than aiming for everything from synthesis like we did the first time around. As this gets a little bit harder and more solid, we want to share those scripts around so that other people can do this too. Again, nothing terribly deep here, nothing terribly sophisticated. I don't want anybody to be looking at that and saying, "Ooh, what amazing science." I want people to be looking at that and saying, why am I editing my things by hand and not just setting up a few simple scripts myself? And I'll end by acknowledging on the designs that I was talking about with these particular examples, there's been a whole bunch of different organizations involved, all the different contributors on the particular project that I used as my working example in this presentation. And if you want to know more about what the iGEM Engineering Committee has been up to, you can check out the site on the iGEM 2021 page. And with that, this is what I wanted to tell you. And if there are more questions that people have, I'll be happy to hear them.
Thanks, Jake. We do have a few minutes if people do have any questions they'd like to ask Jake. You can raise your hand, type it in chat, shout it out. All will be accepted. It must have been very clear.
All right, I'll stop sharing. So that
hang on, there's a question. Do you take enzyme cutting sites into consideration?
That is one of the things that I talked about us wanting to add in. We know that we need to. The first particular run that we did through this, we were synthesizing everything rather than doing a build. The next run that we're in the process of setting up right now, we're going to be doing Golden Gate based builds. And so there we actually will need to be taking enzyme cutting sites into consideration. And what that's going to look like is actually really simple. It's that we're going to add one more link in here, which is that between calculating sequences and exporting to FASTA, we're going to put a little checker in there that just runs through the sequences and looks for illegal cut sites and checks to make sure that the junctions where we want cut sites have the right cut sites there. So we're thinking about this like a software linting tool where you do simple automated design checks. Again, nothing sophisticated, nothing deep, simple to implement. We just happen to have not put it into our particular workflow yet.
Thanks, Jake. Any other questions?
May I?
Please.
This is a wonderful workshop. I'm glad I joined this one. So I actually need to call for another meeting. So I just wanted to ask one question about iGEM engineering. Could you kind of elaborate a little more? In that way I could kind of teach them, kind of learn more from this effort. That's a wonderful opportunity, I guess.
Sure. So let me just go back to the mission piece here. So, and again, I'll recommend go take a look on the iGEM site.
Actually, I'm looking at right now.
Yeah. So there's an engineering hub with a whole bunch of stuff there. So one of the major things that the committee has done is putting up a bunch of webinars on different aspects of the many different engineering challenges in design, build, test, learn that are common ones that people face. A second major thing that the committee provides is fluorescence and OD calibration standards for plate reader and for flow cytometry. So recommendations and software for turning your flow cytometry data into equivalent fluorophore units so you can get things almost to molecules. And for plate reader, there are protocols up for doing the same so that you can get molecules of fluorophore per cell out of a plate reader rather than arbitrary units, and so that you can turn OD into cell count. Nobody should ever use arbitrary units or relative units again. You should always turn your data into equivalent molecules per cell, because then you can relate it to the biology.
Sure, exactly, yeah.
And this last one about the work that we're doing on devices, that's more forward-looking. The big problem that lots of people have in iGEM, but actually in all of our labs too, is fundamentally we want to hook up a promoter to a device, to something else, and have a good prediction about what's going to happen, and be able to take the projects that other people have done and put them together. And we are doing a number of experiments to try to get some areas where that's going to be much more predictable in many people's hands. So the same sort of thing that Chris Voigt has done with cello, only for a wider range of things, and in a lot more people's hands, with units that they can directly compare to molecular models.
Yeah, that's fantastic. Yeah, thank you so much. I mean, this is wonderful for young folks. I mean, like iGEM, that's wonderful. Thank you.
Awesome.
All right. So next we have Gonzalo Peña, who's going to talk about a data management workflow for genetic circuit characterization experiments. So Gonzalo, the floor is yours, and we can see your slides.
Okay, let me do this again. Hello, my name is Gonzalo Vidal, and I will present you Flapjack, a data management analysis tool for genetic circuit characterization. So Flapjack is a data management and analysis tool for synthetic biology. It aims to complete the SBOL-based open source DBTL lifecycle. Flapjack centers on connecting the test and learn stages, collecting and storing experimental data to make it accessible and machine readable. The software consists in a relational database and an analysis engine as backend, accessible through a user-friendly web app frontend or through a REST API. The frontend provides powerful querying and plotting and analysis tools. Everything starts with a synthetic biologist getting experimental data. We have created a semi-automated pipeline that uses the plate reader output. To create structured data from our raw data, we need to provide some extra information. To do this, we rename the sheet that contains the data in the Excel obtained from the plate reader. We also need to add the following sheets: media, strain, DNA, and chemicals. We will fill these sheets with the content of our 96 well plate. The data is structured using the Flapjack data objects, which are the following. Study correspond to a set of experiments or essays addressing a particular hypothesis, like a paper or report. Assay correspond to a set of experimental measurements, including controls, replicates, and different conditions. Sample correspond to the basic unit that is subject to measurement. In this regard, sample can be a well on a 96 well plate. An assay can be the whole 96 well plate, and a study can be a set of 96 well plates. Measurement corresponds to the value of the raw measurement recorder from a particular sample during an assay at a particular time. Vector is the set of DNAs encoding genetic circuit where the individual DNA links to part composition and sequences using SynBioHub URIs. Media is the composition of the substrate that drives the genetic circuit. Strain is the chassis or model organism that codes the genetic circuit. Supplement is any chemical added to the media, and signal is the subject of measurements. For example, a fluorescent channel with given filter bandwidth. Here is the whole data model. We can appreciate how the high complexity data objects are composed by simpler ones. As an example, we can have two plasmids, each one corresponding to a DNA object. Both compose the vector. And this vector is transformed into a strain, and a fluorescent GFP signal can be measured on a sample or well with a media, with a determinate supplement or chemical, which can be a chemical like IPTG. The whole 96 well plate with samples will be an assay, and the assay, a set of assays may be a study. Flapjack has a web app frontend built on React. Flapjack has 4 pages: home, browse, login, and when logged, you will unlock the view page. In the login page, you can register for free or log in if you have an account already. In the browse page, you can browse the data sorted by study, essay, or vector. In the view page, you can query, analyze, and visualize data in multiple tabs. So this is basically the function of Flapjack with his front end, and now we will go to see the function of the Flapjack API. So now I will show you the. Do you see my screen or no?
Yes.
Do you see the color?
Yes.
Okay, great. So here is how we can use the Flapjack API. In the setup, we can install Flapjack and then we import it. With this, we can now log in into our account in Flapjack and import the data objects necessary to work with it. Now we will reproduce the workflow of the web graphical user interface. First, we will go to data exploration and visualization. For this, we will use data from the PAAA plasmid, which is a plasmid that express constitutively CFP, RFP, and YFP driven by the promoter J23101 on the 3 of them. So for this, we call the study named contest effect. We call the vector PAAA in the stream MG6055 set 1 in the media M9 with glucose. To compare between measurements, we can group them. And we will normalize with min max. We will have a subplot vectors and as marker signal. We will plot the mean. And we will get this one. So these selections that I made here are indeed, I import the objects and then pass it to the function plot of Flapjack in this part. And I get this plot. And for example, this is the mean of my data, and I can go and plot the data. See? And with this, I will get all the data points on my experiment. We can go further and analyze this data. To do so, I will plot the PAA expression rates. Here, I will select some sections more will be the analysis type, will be expression rate direct. And for biomass signal, I will have the biomass ID that I export, which will be the OD. So I do that, and I have the plot of the expression rate. But it has a weird peak here that I doesn't notice here, because the expression rate is just the derivative of this curve. So for me, it looks kind of straight. So I expect it to be constant in this part. So I can go further and analyze it using other algorithms, such as one that we are developing, our newest advance and characterization using inverse problems. And now the type of the analysis will be expression rate, but inverse. And now I have a plot like this that will give me the expression rate. And now it seems more consistent with the raw data that they have here. And it's also consistent with the peak of in OD. We'll have a peak in the reporters. We can also lay out this with Flapjack to have it ready for publications. And we can also do repressor characterization in Flapjack. So now we will select decas inverters, all the vectors in this study. We will not normalize. As subplot, we will select signal, markers will be vector, and I will put all the data. This experiment is basically the promoter PLAC-I driven the expression of DCAS9, and the promoter PTET driven the expression of the SMILE guide RNA. Both of them repress the expression of promoter A2, which driven the expression of Venus. And as control, we have the constitutive expression of turquoise driven by the promoter J23101. So here I import all the data objects using Flapjack. And now I will use the function of plot to plot my data. I get some nice induction curves or regression curves. And as well as before, we can lay out in another format, more like paper type format. After that, we see the nice curves, but these curves are not really informative. So we need to parameterize these curves in order to obtain usable data for us. And we will do so with this script here. We will get all the parameters given the following equation. We will have here, we will have the time average background corrected by fluorescence. A is the maximum expression level. B is the minimum expression level. K is the switching concentration of IPTG. And N is the Hill coefficient or cooperativity degree that determines the sharpness of the induction. We get a data frame with all these parameters using Flapjack. After that, we can extend the uses from the graphical user interface to the and use all the power to be in Python doing this. So we will import the contact effects study, and we will get the expression of all the fluorescent proteins. And after that, we will get this kind of data frame, which is really difficult to plot. But now we will use a pivot function, which is a panda property of the data frames. And we will select that the values will be expression, the indexes will be strain and media, and in the columns, I will have the vectors. I run this script and I get a heat map like this. So I will do it a little bigger to show you that now we have strain and media as the indexes, and we have as column the different vectors. Just because using the pivot function, we go from this data frame to a data frame that looks like this. Also in Flapjack, we can integrate SBOL. To do so, we import SBOL and we go to PartShop. With this, we can retrieve information from the vectors from SBOL, from our instance of SynBioHub, and add it to the data frame. So now we have data that has a context, like what was the RFPTU when I was measuring YFP and so on. So this improved the quality of the information that we get with Flapjack, and we are working to extend the integration between SBOL and Flapjack. Then we can integrate Loica, which is a Python package for genetic circuit modeling and design. And with this, we will model a representator. To do that, again, we import Flapjack data objects to work with them, and we construct the genetic network or genetic circuit. For that, we call the genetic network, and we have 3 regulators like I, TET-R, and CI. We add them to the genetic network, and we have 3 reporters, CFP, YFP, and RFP. After that, we will have 2 sets of operators. One is the functional one, and the other will give us a measurable signal. Here we have 3 NOT gates that will input, which input is CI, and output like I, which will replace the following one, which input is like I, and output TET-R, and this replaces the following one, which input is TET-R and output CI. So this driven the dynamics of the system, and this part give us measurement of the system. So this has as an input CI, and as output synthetic fluorescent protein, as well as the following one, each one for the different inputs. We set up the assay, and with this, we create a metabolism and add it to our sample. With a circuit, we select the representator, we have our metabolism, select the media, and a strain. With the sample, we can assemble the assay and run it. Once we run it, we have this kind of data frame, which we can upload into Flapjack. Once uploaded into Flapjack, we can call again our function plot to have a plot like this, to see the behavior of the representator that we just simulated. Again, we can show the different layout that is more for journals, just in one line of code. Also, we can generate logic gates like an OR gate. For this, again, we create or get the data objects from Flapjack. We construct our genetic network. We will be now a NOR gate, which inputs will be AHL1 and AHL2, and this output will be synthetic fluorescent protein 1, which will be a simile of the CFP. We set up the assay, and now we use Python to provide a log space of the concentration of the AHLs, like 8 points from the -3 to the 3 in log space. And doing that, I run the assay and upload into Flapjack. And now I will visualize it using Flapjack. And here it is. So let's see if in the live demo it works. So now I go to study and look for seed. In seed, I click Loica NOR seed, and this will autocomplete all the following sections of the query. The assay will be Loica NOR, the vector will be NOR, strain is Loica strain, because this is synthetic data, and the media is Loica. In the signal, we have CFP and OD. I don't want to see the OD, so I just select the CFP. On analysis, now I will get the heat map. I will select AHL1 and AHL2 as the chemicals, and as function, I will have mean expression. I will plot it with 2 standard deviations and 0.05 as minimal biomass. I select the biomass that will be the OD, and now I'm ready to plot it. I wait until Flapjack does all the work, and I have the nice heat map that I just showed you before. And with that, I am finishing my presentation. If I have more than 6 minutes, I can show you a video of the front end.
You have time, Gonzalo.
Okay, so now I will share the video of the front end. Let me see. Where it is. Let me see if I found it. Too many things opened. Let me just go to share desktop 1. And now I have this. And did you see the video now? Did you see it?
Yeah. Yes.
Okay.
Is there supposed to be audio? Because we're not getting any audio.
Ah, okay. Really? How I can share the audio of this?
You have to, when you share the screen, you select that you want to share the audio.
Okay.
So you'd have to unshare and share the screen again. Or you can do like I did and talk over it and explain it yourself.
Let me pause this. Okay, I will share it with the audio then. Let me see. Desktop 1, share sound. We should be set. Is this okay now? Did you hear the audio?
I'm not hearing any audio.
Okay.
Oh, wait.
Did you hear it?
Maybe not. Sorry.
Okay.
Maybe you'll just have to explain it.
Yeah, I will explain it now. So let me see if I can go through it and without the audio. So basically in Flapjack, you have a lot of different features. I will try to follow this. And we have the browse. You have different studies that you can search. You can search by essay or vector. And finally, we have the view page, when you can view analyze the data. We can informating data. Flapjack allows you to upload data, upload directly from your output from the plate reader, as well as I showed you before. When it has been done, we need to do the extra sheets that I show you also, that will indicate information necessary to Flapjack to build the data objects. Here we can see all of these correspond to the 96 well plate wells from A to G, from 1 to 12, and they have with glycerol, for example. Then, as well as I said, we fill it with the proper well recipient or the media. If you don't have it, you just need to type none. DNA works in the same way. If you have more than one DNA, you need to make a a separation between the different 96 well-played layouts to make Flapjack, or to allow Flapjack to read them. Now we go to uploading data. We must log in to do it. And in the username, we have the button upload. We will have a new tab here. We can edit studies or create new studies. We need to fill the different parts here, the name, the description, and the DOI if existed. We have automated this for HTX Synergy, DMG, and for Flowby. We need to assign a name to the essay. And then we must associate the data with the DNA. So in this, Flapjack recognizes the different fields. And then you can see if your DNA is already on Flapjack or you need to upload a new one. And data upload successfully. Then we can manage our studies. In the browse section, we have, if you have the permissions, it will prompt the manage section, and we can share it with different people or other users of Flapjack using their mails. Or we can make the study public. With this, all people will have access to your data. And also, once public, you can make it private again if you need it. Also, you can delete your data if you want it. But you must be careful with this option because you will not be able to recover the study. Now, viewing a study, we go to the view page and make a new tab. We can change the name of the tab. In this example, contextual effect. We select the study that we want to analyze, and the query should autocomplete with all the parts that are used by that study. In that sense, you can delete some of them, or you can add more if you want. You can also combine different essays from different parts, or look for all the studies that have a particular DNA and plot that data. In the plot option, we have the normalize option, subplots, line, or markers, or plot. For this, we select min, max, vector, and so on. And we have this plot that I also showed in the API. To perform analysis on it, we can go to the analysis section. We can go to expression write direct. We need to select the biomass signal. And now we normalize, mean and standard deviation. Another analysis is a kymograph. Now we will use a different study and select the kymograph section of analysis. We select the chemical that we want to see and the function. Then we plot, and we have the kymograph with time in the y-axis and the concentration in the x-axis. And we can download it in paper format, PNG, or the data as JSON. And then we have different options to format our plots in the user interface. And with that, I finished the presentation of Flapjack.
Thanks, Gonzalo. Are there any questions in the last few minutes? not, thanks for the very clear presentation, Gonzalo. I think it was very.
I'm sorry for the video. I was not
No
prepared.
problem. I think it's better to talk over it anyway. I debated the same thing with my presentation about whether I wanted to talk over it or replay the audio. So our last speaker in this session is Bryce from SIFT, who's going to be talking about a test and learn workflow for high throughput laboratory science. So the floor is yours, Dan.
Great, thank you. Am I coming through on my slides?
Yes.
Great. So thank you, Chris, and thank you to the audience for coming to hear about the work we've been doing on the SG2 program as part of its SG2 stands for Synergistic Discovery and Design. And one of the goals of the project is to better connect data scientists and experimentalists and trying to create a common understanding of experiments and break down the barriers between the people analyzing the data and the people producing the data, and making sure that there's a shared understanding of experiments. So what I'll be talking about is a particular part of SG2 called the round trip, where it's a software architecture that is aimed toward the test and learn phases of a design, build, test, learn loop. And it involves several different organizations that I've listed their logos here on the slide. And if you want to ask more questions, my email's at the bottom, and I can happily direct you to anybody at any one of these organizations that can give you more than enough information about what we've done. But I'll just give you an overview today. The goal that we were trying to address is that try to remove as much of the ad hoc communication that happens between various stakeholders in the experimentation process in order to better capture all the aspects of running experiments, and to ultimately speed up the process of generating high quality data sets for experiments. And a lot of the problems that we had prior to developing the software was where we have people communicating and trying to develop a shared understanding of the experiments. And they do have digital formats for everything that they're trying to specify, but whenever there's an error in the metadata, or there's something that changed, or we had some sort of miscommunication, we have to go back to all these files and update them. We might miss things, and it just becomes a real headache. And I'm sure many of you might have experienced this as well. So this is kind of what I was trying to capture above with this interaction between an experimentalist, a laboratory technician, and a data scientist. They're all trying to collaborate to conduct an experiment that I've shown an image here of the laboratory that we used. This is at Stratios. This is a robotic work cell. So even though there's a high level of automation here, there's still a lot of human manual processes involved. So what we did with the round trip is we built sort of two layers to interacting with the laboratory. The first is a human interaction stage where we're building up the metadata and the description of the experiments that we want to run and visualizing the results. So this is really where the humans should be at, where they're trying to explain what they want and then getting back processed results. And so the automation, what it does is we take their experimental requests and attach lots of information to it and provide tooling that's necessary for them to capture everything in a very high level of detail. And then even after they've attached a lot of that detail, there's still the matter of how do you implement this experiment on a automated laboratory. So we have planning technology that we've used to take the high level description of what the experiment ought to generate and then figure out how to actually implement it using the robotic work cells and the automation available there. And once we've conducted an experiment, then we have several different steps where we do acceptance checking on the experiments. So often what'll happen is laboratories will produce data that might not match what we were expecting. And so we've developed tools to help us identify those mismatches and address the problems as they arise. So once they've been accepted, we have ETL processing. And then following that, we have a very useful set of tools which does data standardization. So from across various experiments, we can compare the data where we have a standard set of columns and values in those columns across lots of experiments, so that we have a standard data frame that we can produce, or a standard set of data frames that we produce. And really that enables the next step where we have several automated analyses that can take those standardized data frames and automatically apply analysis to those, and then ultimately generate a set of results that goes back to our experimentalists. And we have different ways of presenting that information to them, either through developing custom notebooks that you can plot the data however you want, or we also have a tool called escalation that'll allow you to look at reports similar to what you might have seen in the previous talk. And across all of this, we also have a dashboard for the experimentalists. They can track the progress of their experiments, because these things do take a number of days to run, and there's also several processing steps involved, so they can keep tabs on everything that's happening. Okay, so this is the high-level idea of what we're trying to provide with Round Trip. Obviously, I've omitted the design and build phase of BBTL, and we've integrated this with a couple different design and build phases, and it really is meant to address the problem of high throughput experimentation. And the reason we focused on this part of it in SD2 was we were really concerned about being able to evaluate the robustness of various designs. And in doing so, we have to explore a large condition space and try to characterize the operational envelope. And so the test and learn part of it was an essential part of that, where we'd already designed and built these things, but we wanted to do lots of experimentation to understand how well they perform in a lot of different cases. So that's sort of why we broke it apart in that fashion. All right, so this is implemented in an architecture that looks like this. So there's a lot of boxes here, and hopefully this isn't too overwhelming, but the idea is we have several different types of data that are passed between these colored boxes, which are software components. And I have listed the organization involved in developing each of these components. This is a large interdisciplinary team and lots of people involved. So what happens is our experimentalists, they'll start by creating an experiment request, basically where they state what they want. This will go through and we'll attach metadata to that experiment using a tool called the intent parser. And this involves creating links to all the constructs in their experiment to SynBioHub. So we have a record of all the strains that are going through in the experiment. We know a ton of information about them based on what's in SynBioHub. Then at that point, we have a structured representation of the experiment, and it might be missing some information that's necessary in order to actually execute the laboratory. So we have a tool here called XPlan that my group developed that plans the experiments for execution in the laboratory. We send parameters to the lab so that they can know what to do in order to execute the experiment. They don't need to know anything about the context, the intent, or anything about the experiment. They just need to know these lab parameters. And then they produce a set of experimental data. And this experimental data is matched against some expected experimental data in this structured request generator, which this is where we are looking to make sure that we get the data that we expect. And if there's a discrepancy, then we can go back to the experimentalist and say, well, you didn't describe this experiment properly, or we go back to the laboratory and say, you didn't upload all the data. And then we can make sure that we have agreement. And then from there, the data gets stored into our data repository, which is hosted at the Texas Advanced Computing Center. And from that data repository, we go through ETL. And this involves using information from a data dictionary that allows us to attach information to the data that's retrieved from the laboratory to the records for all the strains that are stored in SynBioHub. And then once we have performed ETL on the data, we have a tool called Data Converge, which was where we take all of the information stored in a database and convert it into a data table. So our database is based on a NoSQL database. So it's really very flexible in terms of what you can stick into the database. And that flexibility ultimately makes it hard to standardize. So then we have this tool called Data Converge that does that for us. From there, we have a tool called pre-computed data table that has a set of analyses that it can run on those data frames. And from there, the completed analyses are just additional columns of those data frames that we can visualize through the tool called escalation. And those results get passed back to the experimentalist. So there's been a lot of emphasis in this work in putting kind of checks into the process where we can validate the metadata, correct it, as well as have automation in the process that allows us to easily propagate updates to the metadata. If somebody specified something incorrectly, they just need to go back and change it, and then we push it through the whole process again to make sure that that change doesn't have to have several manual touch points. And what we found is that this has led to a big improvement in our workload, where we don't have to be looking at lots of files. We really just have one place where we do that. And so I'll show you an example of that in a second. This is just a screenshot of our dashboard. We have every single experiment. We have a link to what's called an experimental request that describes the experiment, as well as all of the intermediate data products and timestamps that tell us when they were produced. We also have our main user interface here is a Google Doc called the experiment request. And so we actually found that using Google Docs was a nice way to sort of get collaboration for free, because we have the ability to add comments. We have multiple editors at one time for the document, and we have the ability to add plugins to the Google Doc. And I'll talk about one of those in a second that we use. But what we've done as part of SD2 is we've developed this structure for the experimental request that includes lots of information that is used to recover, like months down the road when we ran this experiment, we didn't really remember why we ran it or what we were looking for, that we can capture that information here in a sort of human understandable prose description of the experiment. And so we have things like the goal of the experiment, the rationale for the experimental method, a point of contact, the date that they prepared it, and so on. And then we have several different machine processable tables in this document. So for example, the one that's being shown here are the experimental controls. The reason we have these in here is that we use them later on downstream in the analysis to know what is our high and low control that we want to use for the analysis. Another piece of information we have in the experimental request is our measurements table. And so what this is each column we're describing, here, what is the control we're using to compare what are the high and low controls we're using. We have a media type, we have a measurement type, file type, the number of replicates we want for each of these, the strains, the time points that we want to measure. This is illustrating a time series protocol. And then we also have a temperature for it. And each of these rows, what it does is it's a compact representation of a set of samples. So I'm saying that I want to run this strain at these 5 time points. And really, if you want to take the cross product of the strains, the time points, and the replicates, that would expand out into a whole bunch of different samples that you're going to generate. So we think of this as a relatively convenient way for an end user to specify what is the set of experiments they want without having to list out pages of tables. And then lastly, we have a parameter table that these are parameters for the protocols. So this allows you to control things like different volumes that are transferred between wells and other configurations, such as whether you're using new induction plates every time that you make a transfer. And it really is a protocol dependent set of parameters that you can choose. But we allow the users to specify these. And this is something that we, because we're using Google Docs, it's been very helpful to have, because we're able to annotate these and add comments to them and have a discussion about these. Okay, so from there we have a tool called the intent parser, which what it does is add several different ability to add metadata to this. So one of the things we can do is we can add links to SynBioHub for each of the strains. It provides some suggestions for these and allows us to link them. And from that point on, we know more about the strains, and we can add new ones if we choose to. And then from there, we can request an experiment execution, which will then initiate a series of automated steps and result in running the experiment at our laboratory. All right, so the first thing that happens after you ask the system to run the experiment is we create something called a structured request. This is really the first place that we've taken the Google Doc and turned it into something that's a formal structured representation. And this, what it does is it captures all of the information about the samples that's provided. And it also incorporates the linkage back to SynBioHub for each of the samples so that we know what it is that's going into constructing those samples. Once we have a structured representation, we use our tool called XPlan. So what XPlan does is it takes the requirements that are expressed in the experiment request. So this purple box is kind of illustrating this is like the things that the experimentalist is asking for. And then we also have what's called a condition space, which is the space of possible samples we could possibly run through the protocol. And then we also have a specification of what containers we have on hand at the laboratory. And what XPlan is doing is it's trying to solve a partition problem of the requested experiment samples and the containers, and trying to make sure that it can satisfy this request given the containers available at the laboratory. So if it can do that, then what it does is it produces a set of parameters that will be used to invoke a protocol. And so some of these are specified by the user as part of the experimental request, and others are ones that are decided by XPlan. So one of the things that it picks is, in this time series protocol, we have an inducer that is addressing each column of a 96 well plate, and it will pick, which column gets what level of inducer. And you don't require a user to tell you that sort of thing because it doesn't really matter. You care about getting your sample measurements. You don't really care how the plate's laid out. All right, so then once we submit the lab parameters, what we get is this is through the Stratios web interface. I'm just showing that we have a series of runs that have been queued as a result of processing the experimental request. And we can go in and look at each of these and see what the instructions are. So here for this time series protocol, involves a series of steps that involve liquid transfers, as well as down the line, we have incubations and various measurements that are being performed on the plates. And at Stratios, they also provide data that's produced as a result of running the experiment. So because of the time series, we get plate reader data as well as flow cytometry data at different time points, and just illustrating some of the fluorescence data that we got from the plate reader for this experiment. All right, so then once the lab finishes the experiment, they take the raw data, which is in the form of CSVs or zip files of FCS files, and then they also describe the data in terms of what's called a sample trace. In this sample trace, I've shown an example here. It just says, for this well in this container, I measured this measurement at this time. And it tells you what the lab actually ran. So at that point, you need to look at this and say, well, is this really what I asked for? And so we can check and make sure that that's the case. It is. We'll go and run through a series of ETL steps. I'm illustrating here one of the steps here. We use TASB to analyze the data. And from there, we have what we think is a complete data set, and we want to make sure that we can then compare it across different experiments. And so we have a tool called Data Converge where what it does is it produces several different types of data frames from both the plate reader and the flow cytometry. So it'll include raw data, log transform data, as well as the ETL process data. And for each of these, it has metadata on the data that it processed. So one of the things it provides is the columns that appear in the data frames and frequency of values reported for each of these columns. And this should just give you a sense of the types of things that we capture in the data. We have replicate IDs, the controls, the media types, the inducer concentration, the time points, the aliquot IDs in the containers when they were run through the experiment, the amount of volume that was used in the flow cytometry, the estimated cells per milliliter, and so on. And an important point about this is that these change over time. So we have analysts that request new columns all the time. And because the way that we've built the system, we can add that to old experiments and just reprocess everything without having to go in and modify our data frames at all by hand. So this has been a really helpful thing for us as we find things that should have been produced in the data frames that they're hiding somewhere in our database. And we don't want to have to redo everything that we did up to that point. So instead we can, because of the fact that we're describing the data in this way, it's very easy to just push a button and bring through that metadata to the very end again and rerun the analyses. Okay, so once we have the standardized data frames, we have several different analysis tools that various people on the project have created. Just to give you an idea of some of these, we have a tool called Diagnosis that looks at sources of variation within various experimental factors. We have another tool called Signal Prediction that takes flow cytometry and tries to predict the high or low output signal from GFP. We have another method for labeling flow cytometry events as live or dead. Another that called performance metrics, where what it does is it calculates the ratio of the low to high signal for our strains that are representing genetic circuits so that we can see the separation between the low and the high. We also have various distance metrics with they kind of do the same thing. Instead of a ratio, they're using Wasserstein distance to compare two different distributions of flow cytometry events to see how far apart they are from each other. It also gives you an idea of like how strong your signal is coming out of your circuit. And we also have growth analysis tool that will predict doubling rates. And because this is built all in a standard data frame or data frame format, it's relatively easy to add new analyses. So there's opportunity to, as you know, data scientists develop new analyses in a notebook, they can easily package that up, deploy it as a new container on our system, and just have it run automatically when a new experiment comes through. Okay, so I want to give you an idea of what some of these analyses look like. So this is from Perform Metrics. And what I'm illustrating here is we're taking each of the conditions appearing in the experiment and drawing a box plot of the high and the low signal for the circuit in each of these conditions. So in this case, we have 6 different circuits that are various logic gates. So we have NOR and XOR and so on. And this can give you an idea quickly whether you're seeing the right distribution of inputs or distributions of outputs for these strains. And this is a similar view from the performance metrics where again, you're getting a box plot, but you're showing the ratio of the high and the low signal. And what this is doing is it's looking at all the experimental factors and trying to look at the distribution across one factor at a time. So here we're looking at each of our media types and we're trying to get an idea of how well we're separating our low and high signal for that particular media. Which would be this orange red box here, would be the example for sorbitol. All right. Another thing that we can do with this that has been useful is it gives us ability to compare analysis techniques pretty easily, because we have a standardized input for all the analysis tools, and we can have several analysis tools that provide the same sort of output. And we can then compare them against each other to see if they're same and they're not completely wildly different. So we produced this analysis here where we have four different ways of predicting the fold change in a circuit based upon the inducer concentration. So it goes from red to blue as increasing inducer concentration. And we saw that between all of these techniques, they had tended to be correlated relatively well. They didn't agree in all cases, but it gave us some sense that the predictions that we were making with these tools were not completely crazy from each other. Okay, so with my last slide, I wanted to just give you an idea of what this has done for us in terms of developing this sort of automation. So what I'm showing on the right-hand side with these two plots are the number of samples generated and the number of experiments run over the course of the ST2 project. So we started around the beginning of 2018, and we're here currently in early mid-21. And I'm indicating the point at which we deployed the round trip to help us with automating experiments. And so the ST2 program is broken up into 3 phases, which I've indicated by the blue, orange, and gray data points. And at the point that we deployed the round trip, we had a bunch of data already that had been produced by other experiments. We had more manual ways of doing this. And what we it took us a long time to get any data incorporated into our database and ready to be analyzed. And we had a big jump where we went from almost no data up to, over maybe 150 experiments. And then once we deployed the round trip, we saw a much more consistent addition of new experiments to our data set, or a set of data sets. And that the actual pace of running experiments was an improvement of about 30%. And similarly, we saw an increase in the number of samples generated as well, or the increase in the pace of generation of samples. So after we deployed the round trip software, we've run about 100 experiments and generated about 75,000 samples as a result of that. And what this ended up doing for us, when we went from this previous sort of manual process, it took us about 3 weeks to do an analysis on an experiment. And when we first deployed the round trip, it took about maybe a week, because there was a bit of manual analysis still done on the back end, where we had to still consult. We had the experiment submission part of it automated, but then the experiment analysis side of it wasn't completely finished. And then now toward the end of the program at phase 3, we've brought this down to about 4 hours to do the automation side of things. Obviously, experiments still take time. They still have to incubate, and that takes a few days. But in terms of the time to do the analysis, it takes about 4 hours. The other thing that we've seen is we've had just probably want to focus on the orange here, the number of data products produced, that because we have all of these analysis tools that are being based on the same common representation, we can produce that many more data products as a result of that. The other thing we've done is we're supporting 5 different protocols that have lots and lots of parameters, so no 2 experiments are alike. And I characterize this as having about 15 different qualitatively different types of experiments, that even within each of these protocols, there's lots of ways to configure it. And there's sort of, I would say about 15 sets of those that we've done that are different from each other. And with that, I will open it up for questions. We have about 5 minutes, I think.
All right, thank you, Dan. Are there questions that anybody would like to bring up for Dan? All right, we've got a question from David. How much of the code is public and where can you get it?
I think that depends on what part of it you're talking about. The whole set is not public. It's all running on the Texas Advanced Computing Center's site. So see, let me go back to the architecture diagram to show you what is actually public. So I think that TASB and SynBioHub are certainly public, and Stratios, anybody can submit experiments to Stratios. I think the rest of this is private.
Actually, a lot of it is on the SD2E GitHub. So I know intent parser and a number of the other things have gone out there. And anything that's not there is supposed to migrate there as the program wraps up. So I posted that link in the chat.
Thank you, Jake. Yeah, that's right. As part of the goal of the program, we're planning to open source, I believe, everything. And I also believe the data sets will be made public at some point as well.
Yes, I believe the data sets are supposed to go public. I'm not sure that I'm not sure that DARPA has figured out exactly how. I pasted the links for the other things that are living in other GitHub locations as well. All right, there's a question from Nathan. How much of this tooling end-to-end uses the SBOL3 data model? Or SBOL or SBOL3, I think is how I'm interpreting the parentheses.
Right. So yeah, this is something that we've been working on. I think a lot of the things that have gone in SBOL3 have been motivated by challenges we've had or that other people have that are similar to what we've had. And one of the things that we've been interested in with respect to SBOL3, I think, is the description of the experiments. And there's, I think, a pretty good mapping between the experimental requests and SBOL3. SBOL is mainly used as part of describing the biological designs. And I think that's it. Jake, was there anything else that you would add that is
the intent parser, the data dictionary, and SynBioHub all are SBOL.
Right.
A lot of this, the things on the right-hand side, mostly don't yet use SBOL because SBOL3 didn't exist when all of this was started getting wired together 3 years ago. At this point, there's actually a slightly newer version of this where work that's being done on protocol representation and requests for protocols is also all based out of SBOL, and some of the pieces in the right have gotten that wired through them now as well. But some things like the actual interface to Stratios is in a custom Stratios API that we have to hit. And so as you get into specific legacy things, you get away from the standards. But I'd say about half the links on this have an SBOL component to them. And
it's been a big effort.
Yeah, there's still threads that lead back to SBOL.
That's right.
All right, we're coming up to the end of the session. Any last questions before we go? All right, I think that is a wrap. Thank you to the other speakers, and thank you to the audience for good questions. And on that note, see you all later.
See ya.